first speaker is Dr. Fred Pryor from Arkansas, giving a lecture on data curation and quantitative analysis, NCIS Cancer Imaging Archive. Fred, thank you very much.
Thank you for the introduction, and thank you all for being here on this lovely afternoon. I will try to keep it a little bit energetic so that I keep you away from going outside where it's beautiful. I think Professor Marty Bonmati and I should have been back to back. We are, but separated by the tea break, because we're actually giving, I think, two halves of the same talk, and you'll see what I mean by that in a second. So I'm really going to focus on the data that we need for developing new algorithms and what it really means to do data curation, what the data quality aspects that are required if we're going to move forward to actually make usable and useful tools. So we focused a lot today on supervised learning, and I'm going to continue to focus on that approach because that's where we need the most labeled data. We need labeled data in order to train the algorithms, but to validate them as well. So I'm going to talk a bit about how do we get labeled data, but first we're going to talk about how do we get the data that we use in order to generate labeled information to train these data sets. To train the algorithms. So my contention is for a machine learning algorithm to be clinically useful, it really has to be trained on data that appropriately represents the variation in human biology, the variance in the presentation of the disease of interest, and the variance in the data collection systems. Now, we've heard this all mentioned already today, but that first one we never pay enough attention to. There are 7.5 going on 8 billion people on this planet, and they're not the same. In order to look at the variance in that population, you need a lot of data. So in the US, we have two projects, the All of Us project and the Million Veterans program, both of which are collecting data on 1 million people. In the UK, you have the UK Biobank program, which is similarly sized. Those are beginning to be the right size. And unfortunately, only the UK Biobank has imaging. We couldn't convince the Americans that imaging was worth the expense. So there are a number of limitations in terms of the data that we have available and limits its availability for use in AI research and machine learning research, but also this limits its availability to the research community in general. I've already made this point about the variance in human population. Well, when we collect data, and particularly the collections that I'm going to talk about that was funded by the U.S. National Cancer Institute, we collect cancer patients' data, not normal, healthy humans. So it's really difficult to train algorithms to look at the variance in the population as a whole when all you have are examples of pathology. Now, there are some cases where there are very large normative data sets. For example, the Human Connectome Project in neuroscience collected 1,200 normal, healthy volunteers. There's a whole cadre of follow-on connectomics projects. So if you're looking at gliomas, glioblastomas, life is pretty good because there are very large, very well-curated standard data sets that you can use for comparison. But for most cancers, such data sets really don't exist. So that is a key limitation that I keep pointing out to the National Cancer Institute, that we really need to be collecting the normals to compare. But there's another interesting problem having to do with intellectual property. So we've talked a lot about algorithms that are being developed for clinical use. Those are medical devices. People sell them. And so in order to get those devices approved, they need to have data. Now, in many instances, the company and the institutions, academic institutions, consider data to be their intellectual property. They want to hold on to it. In fact, many researchers have that idea that they do not want to share their data. It's part of what they've generated as the intellectual contributions for the laboratory. Dr. Kalpathy-Kramer mentioned challenge competitions. We participated in a number of those, but providing data, that's a nice way to develop algorithms in a competitive environment where you might win $1 million if your algorithm does better than everybody else's. But really, from my perspective, it generates labeled data that we can then capture and reuse for other purposes. But it also means that we sequester data during the competition, and in many cases, we sequester that data forever, which takes it out of the pool of useful research data. Now, the FDA has argued very strongly in the US that they must have sequestered data in order to validate algorithms that are going to be used commercially. And there's a lovely publication that just came out, I think last month, there's a reference to it, which describes the FDA's overall framework for dealing with software as a medical device, that's what that acronym means, that are based on AI and machine learning. So the FDA has spent a lot of effort on dealing with this problem because they know it's this massive freight train that's coming right at them that has to be dealt with. They have to have a framework in which they can deal with the information. So I'm not going to go through all that, although it's a great document. I think they've done a good job. There are different classes of medical devices in the US, and depending on what class your device is, it may need to actually go through the complete clinical trial or a 510K or even a simpler process. But what I want to focus on here is the amount of data that this process takes out of circulation. It's a sequestered data that, of course, is needed for model validation. The FDA wants to control a data set. They may reuse it to test multiple algorithms, but they want to have a very large labeled data set that they're keeping. Of course, the vendor has gathered a set of data that they use for training and testing their algorithm. And this model actually takes into account this retraining approach where you put it into the marketplace, but the algorithm continues to learn. So that's basically your data from your institution, but both the vendor and the VA want to control that data. So we're now talking about a very large amount of high-quality data that would be very useful for research that's locked away, and you'll never see it. We'll never be able to use it for research. So this intellectual property problem is sort of normally invisible, but we deal with it all the time as we lose a lot of data to this process. So several people have also mentioned the truth problem. And I think this is a critical issue. We have no ground truth. I remember when I wrote my very first grant dealing with quantitative medical imaging, and I said, well, okay, so what does a radiologist use for truth? Well, what the pathologist said. Okay, so I looked at what the pathologist problem and said, well, what does a pathologist use for truth? Pathologist is always right. This didn't sound to me like a good plan. We don't really have a good gold standard of truth. But what we do have is a number of ways of approximating that, and one of the approaches that I think is most useful is this idea of taking multiple observers, they may be human or machine, and finding ways of weighting their contribution. In other words, an estimate of how accurate their information is, and then coming up with a voting algorithm that combines them. So there are two algorithms one is very well known, the staple algorithm that Simon Warfield at Harvard came up with some years ago. We made a little contribution to this, an algorithm called Veritas. They do pretty much the same thing, although we have different standards. But the point is, we don't really have ground truth, but maybe we can model truth. And in order to validate algorithms, we need some consensus models for what truth is. Okay, so we've heard a lot of people talking about data, and we need a lot of data. And I've already hinted that I think it's the scale of data based upon the variance in human population. But there's another way of looking at the amount of data. Deep learning algorithms, and you've heard a lot about that today, had this really nice property that they don't necessarily need the input of, say, what a radiologist is looking for in the images, the engineered features. They can learn their own features, but they need a lot of data in order to do that. And so Ian Goodfellow, who works for Google, wrote a wonderful textbook on deep learning. And in it, he has this nice rule of thumb that I use to guide my thinking on this. And he says, whoops, a supervised deep learning algorithm will generally achieve acceptable performance with around 5,000 labeled samples. And you saw several speakers today present data that sort of corresponds with this. Between 5,000 and 7,000 cases, you seem to get good results. But if you want to get over this issue of generalizability, if you really want to represent the biology accurately, it's more like 10 million. Well, if you Google, that's easy, right? I mean, if you're trying to figure out what I'm likely to buy next week when I get on the internet, there's tons of information about me, in fact, about all of us. So for them, 10 million labeled data sets is a small data set. But for us, that's a whole lot of data. And how do we get that amount of data, make it available, and more importantly, how do we label it? So that's really what I'm going to deal with. And since 2010, my group and I have been dealing with this problem of how do we accumulate information and make it publicly available. In 2010, the National Cancer Institute in the US put out a request for applications to build an information resource, a cancer imaging resource, and to maintain it. And for better or for worse, we won that and have maintained it ever since. It's called the Cancer Imaging Archive for an interesting historic reason. The Cancer Genome Atlas was being built at the time. TCGA sort of made sense. TCIA doesn't make sense, but at least it was better than CIA, which is the other answer. This is information repository that's built on what's called the FAIR principle, which is that data has to be findable, accessible, interoperable, and reproducible. We work really hard on the first three. The fourth one is sort of up to the people who give us the data, but we do our best to make sure that it's at least properly discoverable and you can cross-link it. And so that's really what we spend our time doing, is acquiring data, both imaging data, radiology and pathology predominantly, although we have a growing number of small animal imaging data sets, PDX models, et cetera. But also information about the human subjects who gave us the data, or the animal subjects, clinical information, outcomes data, and a growing amount of genomic profiles or proteomics data. Too much pathology in the mind. So it's a very large repository of a wide variety of different types of information, and the key thing that we spend our time doing is curation. And that's really what I want to talk about, is how do you make data useful? How do you make it findable and interoperable, et cetera? So this is just the sort of the eye candy slide of the kind of information that I've already mentioned: pathology, radiation therapy data, a huge amount of that, and then all types of cancer imaging information. It's an information repository that you can search online. About 15,000 people a month do that. They download on average about 75 terabytes of data a month. Last time I did the rolling 12-month average, which was a couple weeks ago, it was 950 terabytes of data every 12 months that are downloaded by researchers from 125 countries around the world. So it's a well-used repository. You've heard several speakers today who use data from TCIA. We keep track of the publications that have used the data, retrospective research using this information. Now, one of the things that to me is key for having high quality data, and I'm going to talk about data quality in a minute, is that we need to ensure that the information that we get has been consistently collected with consistent protocols. I did an experiment when I was at Washington University where we had collected data supposedly with a standard brain protocol for looking at GBMs, and the short answer was out of 300 cases, I could find 11 that actually consistently adhered to that imaging protocol. Now maybe it's better in the UK and other places, but in the US, clinical information is not gathered according to consistent protocols. You can define the protocols, but the data is not collected that way unless it's a research trial. And so we spend a lot of our effort focused on collecting data from high quality clinical trials where someone has spent the time to look at and enforce rules about the acquisition protocols, and they've done quality assurance on the images, at least to some level. So that's the first stage, if you will, of assuring high quality. Now, of course, it's already been pointed out that there are a limited number of such trials, but as you can see from this number, these are the groups that we're currently working with in the US, and in the next 3 years, we're looking at basically multiplying our data volume by about a factor of 5. Now, a lot of that is pathology. They grow very, very quickly because pathology images are quite large. So this is how one of the key approaches that we're using to gather a large amount of data, having that data be of reasonable quality so that we can then, through our curation process, make it cross-linkable so that we can take data from multiple lung cancer trials and link it together into meta-analysis sets that we believe can be used for machine learning algorithm development and validation. So what do I mean by data quality? A good friend and colleague of mine, John Talbert, actually runs an entire academic program on information quality and data quality. I'm not going to bore you with the difference between the two because I tend to focus more on the data quality, but he has these 4 pillars. Data quality is based upon data governance. Now, a bit redundant here, data quality, but for me, that really should read data curation, which also covers data integration. How do I make sure that the data is cross-linkable and that all of its internal linkages are correct? And then finally, analytics. So for the Cancer Imaging Archive, we have a rather complicated data governance process that starts with, if you want to give us a data set, you fill out an online form and it goes to the National Cancer Institute, where a committee decides whether or not they're sufficiently interested in your data set to warrant its collection. That tends to filter out a lot of data, which I would like to collect, but it is the process that our government has given us. But it does one very important thing for data quality. The National Cancer Institute makes sure that all of the data was collected with proper consent, so that the human subjects consented for the reuse of their data in an anonymized form, and that the data was collected according to the appropriate rules governing the use of humans in research, depending upon the country in which it's gathered. We gather data from all over the world. So that's their job, and I'm very happy that they do it. We have an institutional review board protocol that governs what we do with the data to ensure that our institution is protected. But that combination is our way of making sure that we have dealt with the human subjects research questions accurately and adequately. And then we have a large, actually I think 5 now, different ways in which you can upload data to us depending upon the kind of data and where you are. And all that data goes into a curation process, which I said before is, to us, the key. We collect data so that it's de-identified before it leaves your site, mostly de-identified. There's always hidden stuff, so that's part of the thing that we do once we get it. But we have a set of tools which we've developed over the years called POSITIVE. They're now released open source in a Docker container, if you'd like to have them. These tools ensure that we've removed all protected health information, that all DICOM data structures are properly integrated correctly, and a number of other things having to do with data quality and interoperability. So that's a set of tools that we use that implements this rather large pipeline of analysis steps to make sure that we have high quality data, which we then store and make available. So the data is not visible until it's gone through this, even though it's been transferred to our site. We maintain it on a private cloud. Our private cloud, because we also manage a repository for the Centers for Disease Control, we are attacked on a regular basis by U.S. Homeland Security to make sure that all of our security protocols are in place. That's not necessarily a good thing from our point of view, but it does mean that we maintain proper security. So those are the first 3 pillars. The 4th pillar of data analytics is something that we're still working on. This is, again, some eye candy that makes the unfortunate point that most of our analytics focus on usage, because that's what the government is interested in. There are people using this. But really, the analytics that are of value have to do with what we do automatically. So all of the data that we collect, we calculate a checksum, and we check that in the background on a regular basis. So there was a question earlier about an Israeli prank of automatic or embedding some interesting images in a public collection. We would catch that through this process and would correct the information. Now, we've never had that experience yet. That wasn't our data, but we have already built in the ability to detect that. There was also another case where there's the DICOM standard, which we rely on heavily, has in it what's called dual personality capability. And I won't bore you with what that means, but somebody figured out that you could put executable code in there, so turn a DICOM image into a Trojan horse. We also, within our POSIT toolkit, catch that and correct for it. There's a publication that some of my people, it just went out yesterday, I think, how to deal with that problem. So those are the kind of quality metrics that we think are important. We don't report them, but we generate them in quality processes. Now, we still don't have enough data. And these are the top 10 in terms of the number of human subjects participating, data sets that we have by cancer type. And you see lung cancer is by far the largest because we have the data from the US National Lung Screening Trial, which is 27,000 subjects. And you'll notice that this is the number of machine learning-based publications or radiomics publications, so it correlates. And in fact, we have a pretty decent correlation. The more data, the more publications, except for brain. There's been a whole lot of work in GBMs, I think largely because of the Cancer Genome Atlas. This is a lot of work is cross-linking the genomics with imaging features, but also because there's availability of normal data sets. Now, capturing labeled data is the real problem here. So far I've talked about capturing data, but now somebody has to label it. And how do we do that? You can do the math yourself if it takes, let's say, 20 minutes to go through a lung case and find all the nodules or a brain looking for all the metastatic disease and segmenting all those features. If it takes a radiologist about 20 minutes to do that, so they can do 3 an hour, and I want 10,000 labeled data sets, that's 10,000 hours. You know what a radiologist makes. So you can guess this is not a cheap process. But I really wanted 10 million. That's a very not cheap process. So there are some alternatives for actually having those expert labels, even though those are the best labels. We've already heard about Dr. Kalpathy-Kramer mentioned crowdsourcing. That is a really important area that I think, as long as you're doing it with, for example, her Crowds Cure Cancer approach. Data augmentation and synthetic data, these are ways of actually generating more training data that we already have a label for one, for a given data set. Now I can morph that data set so that, for example, it looks like it was generated on a Siemens machine when it was actually generated on a GE machine, because I don't, but folks at Harvard have models of those different machines, and so they can generate a data set as if it had been generated on the other vendor's machine. That kind of data augmentation, I think, is very important to expand our labeled data cheaply. Jayshree's already talked about this. I think this is a very important approach to labeling data because you get hundreds of people, most of them radiologists, labeling the same information. So now I get statistics. I don't have to worry about, do the 3 or 9 radiologists agree or disagree. I have 100 radiologists. I have a statistical distribution. That's much more satisfying to me, for me to work with. But what really is labeled data? This is something that we've been struggling with because we want to curate and manage labeled data. So what is it? You can start with outcomes. I purposely picked pathologies because there's a room full of radiology people, so I'm going to force you to look at pathology slides. But it's the same problem, right? I can start with outcomes. So if I'm doing a deep learning algorithm, maybe all I care about is which subjects have cancer, which don't, or which have glioblastomas and which have gliomas, low-grade gliomas. So outcomes data might be enough. But most of us are worrying about segmentation. I have to identify objects in the image and segment them. Well, if you're talking about lung nodules, you might have a dozen. That's a path slide. There are 1 million cellular nuclei in there. Human beings will not segment a million cellular nuclei. It does, full stop. So that has to be done by an algorithm. But is that really what I want? Or do I actually want the features that are derived from that, the mathematical representations of the characteristics of those objects? Or do I want clusters of that information? Because that's really what's more interesting for the algorithm that I'm developing. I could represent those clusters, in this case, as tumor-infiltrating lymphocyte maps, and I might want to compare different TIL maps. So what is a feature? What is the label that I really need to keep in my data set? Well, the answer is all of the above. We've been working with colleagues at Stony Brook, Joel Saltz and his team, on the pathology version of this, hence the pathology start. All these tools are part of the Cancer Imaging Archive, but we're focusing now on this one, which is a feature base. And we're trying to develop the standards for how do I represent image features, because they don't exist. There isn't a DICOM standard for image feature sets. How do I represent a feature vector? And what we've determined thus far is, in fact, the most basic format in which to store this information is a fourth order tensor. Great, compact mathematical representation, really hard to structure for a database to manage. So we're struggling with how to organize this information and how to manage all those classes of features, provide some kind of semantic integration. For engineered features, there are ontologies which are growing and are very good, but for learned features or for annotations, there really aren't. So, and then how to link these features to the data that they came from, because we combine information from multiple trials. So the images, a given feature set may have come from images from different places, and we need to maintain all those linkages. So my summary is actually very similar to what other people have said, which is we really have to have high quality data. I think I've given you my definition of high quality data, which is a little bit different. We have to have huge quantities of data that is labeled. I don't think that human labels are going to do that. And as I should have mentioned on the point I made about the Staple and Veritas algorithms, human labeled data for machine learning is a trap, because if the algorithm does better than the human expert, you will always be penalized, because the human expert was the definition of truth. So we need models of truth that don't penalize the algorithm so that they can actually produce better results, we hope. And to me, the answer is open access repositories that allow us to all share this information. And as I've shown you, we've been pretty successful at building such repositories and having people share them. Thank you. Oh, sorry.
The next presentation is on the role of open source and open collaboration for imaging AI, and it will be delivered by Dr. Ki Moon from Arlington.
Thank you very much. Oftentimes when one gives a presentation, sometimes one ends your presentation with the announcement that we're having such a conference, please come. I'm doing it backward. Last week, I worked with Fred Pryor to organize a small workshop with 50 people on a very similar topic. I started the workshop with these two cartoons. Is AI gonna make us better or worse? But the most interesting thing was the one about the class on machine learning. One thought, wow, machine learning is really exciting and interesting. If I do that, I don't have to deal with data normalization. Well, you cannot escape that. And we are learning that over and over here in Lisbon. First, I was puzzled by the title of your conference. It's not a word that we use that much, at least in my circle in the United States, so I looked it up. It has different meanings. So I'm trying to focus on the second part of the meaning, trying to understand what this widget is all about. There was a workshop hosted by NIH last year and published a paper. And in that paper defines machine learning system must be capable of explaining or illustrating the advice they provide to human users. So this issue of explainability becomes an important part of the discussion. But in that, we also find, we hear about black box all the time. Then recently, there was a report published by a group of radiologists, and the document looks like a number of attorneys participated, talking about ethics of AI in radiology. I'm not an ethicist, but this is your AI. Something goes in, something comes out. In recent years, there has been explosive growth in publication of use AI outside of imaging, medical imaging community. In those cases, you see that they do a reasonably decent job of producing something, but not that relevant for medical application. So for those of us who are working in software, sometimes we talk about open architecture, okay? That is, we need to see what's inside. And when you go to AI meetings, you see this type of diagrams all the time. So in a way, this describes different parameters and processes in the AI process that you can control. But oftentimes, we don't really know what really those things really do. Now, you radiologists will not need to know beyond this perhaps, but your research partner who, on the technical side, will need to know more than this. So this is where the open source software comes. The software, completely open, will give you a maximum transparency. And also what you will learn that some of this open source code that you can download from multiple sites are not necessarily optimized for diagnostic imaging purpose. So why am I talking about open source in machine learning or AI conference? There are increasing number of open source machine learning packages all around us, perhaps even 100 or more. And many papers, especially outside of medical community, have been published using this black box and say declare victory. Now I'm sure some of you here will use some of these open source packages in your research. And also, I'm sure our industry partners are looking at a lot of these open source packages as well. So what do we really need to know about the best practice in open source? And how do we take advantage of the open source software community to advance your research or development agenda? Open source concept got actually started quite some years ago in the early 80s. And in some ways, open source is very misunderstood because open source is readily available free of charge. So it's a free software, Yes, software may be free, but understanding the software is not free. The free has more to do with the freedom of what you want to do with the code. In a way, it's a social movement and philosophy for developing software code in a shared environment. Now, this sort of open source concept got codified a bit further because some years ago, the American government said to AT&T phone companies, you know, you're in the phone business, so you should not be developing software. So in order to force the AT&T just to do phone business, they forced the AT&T to release a number of their intellectual properties to the community. So one of the things that was released at that time was earlier version of Unix operating system. Under the stipulation that the software can be licensed without royalty payment, that is no fees, under no obligation for technical support. So AT&T does not have to be responsible for the software, but you cannot charge any money. Therefore, it became the beginning of concept of free software. But understand, the free software has no particular obligations to the end user. Then, the software package that came out of AT&T went through a number of iterations in the community. So Linus, wrote additional packages to make the Unix operating system that much easier to use. So eventually, that became one of the most popular operating systems in the world. And then also, Linus is the guy who developed the very first version of Git. That has become GitHub, and now it's owned by Microsoft. Many different companies have open source business, open source policies, or some organizations, their entire business is open source. You have known Linux Foundation, and there's an organization called OpenStack. This is a software package that came out of NASA to the public and became the core technology for most of the work you do in your cloud environment. So you see a number of different open source activities like that, but they all use different open source licenses that supports their business model. Yes, it's free. Yes, it's readily available, but there are certain conditions, and those conditions are the ones that you should be familiar with. Now, when I started this open source foundation about 7 years ago, with the sponsorship of the United States government, I would go give talk about open source, and mostly companies, at least in health IT, you're a very dangerous guy. Open source software is dangerous, unreliable, and shouldn't be used in healthcare. But that's not the first time I was told being a dangerous guy. In the late 80s, I was very much involved in promoting PACS with the huge funding from Department of Defense. Again, I was told I was a dangerous guy promoting digital imaging. Number one, image quality will never be good enough. Guess who said that? Kodak Corporation. Have you ever heard Kodak Corporation? Then I go to radiology community. You're a very dangerous guy. You're going to make radiology service a commodity service. Well, today, earlier I heard that somebody reported PACS really has improved efficiency of radiology service by 30%. That really makes me feel good. So I was not that dangerous. So a little bit about open source in terms of more legalistic terms. Open source software has to be licensed under proper license. The creator of the code has the copyright, and the copyright holder issues a license so that the end user can study the code, change the code, and distribute the code following the rules of the license. The two things are important here. Without proper license, it's not considered as open source, okay? And also copyright holder, that could be the individual or the organization, provides rights to license. So if you look at the code, open source code, there has to be a sort of header information like putting in the beginning what this license is all about. There are many different licenses, maybe 50 or more. Then these licenses are not equal. Sometimes they're not even compatible. For instance, if you're in this space, if you see the license with the GPL license, and if you mix this code with the license from this code, they get all mixed up. We call it extremely viral. The license on this side is very restrictive. License on that side, it's very flexible. So at OSERA, my organization, we support Apache 2 license for all the things we do. This is not to say you shouldn't use this. This is not to say you shouldn't use that either. There are rules how you should mix and match. And also you can relicense it. You can get a license, Apache 2 license. For whatever reason you'd like to make it more restrictive, you can relicense it under this. But if you start from here, you cannot go back there. Now, in the past, this GPL license was much more popular, but over the years, you see that more use of the Apache 2 license, more flexible license. Whenever we think of open source, and somebody briefly also mentioned the open data, we think of the computer software itself. Yes, that is true, but there is a large amount of activity goes behind. So the ecosystem of open source includes certainly the software, rules of engagement, business rules that has to do with the license, and the community. In fact, the community may be the most important part of this open source phenomena, so to speak, because that's the community you interact, and that's the community you collaborate, and that's the community that can help you if you're willing to contribute. And open source code can be commercialized. As I mentioned earlier, code itself doesn't have any warranty. Sometimes it doesn't even have a user manual. But if you want to productize it, make a commercial product, you can do so by adding additional services and capabilities. Back to machine learning. These are some of the popular convolution neural network packages you can find on the internet, and the number is growing. So if you look at some of these packages, there's a list of creators. The first one is out of Berkeley, and so on and so forth. And some you can see are supported by very large corporations like Google. Then it has certain licenses. You can see the BSc license, MIT license, and so on and so forth. Then the platform. And also it shows when it was released, and also it shows what language that was written in. So your technical partner might have a preference over one language over the other and one platform over the other. So that will be one of those factors you need to consider making decisions. Okay. And here you can see a very high-level summary of different attributes of different licenses. And also there are some additional details that you can look up. And in this case, for instance, TensorFlow is one of the more popular, more sophisticated. However, if you go to Keras, or that is less flexible, but it's easier to use. So if you want to start something fairly quick, perhaps you might want to look at Keras or Caffe 2. For instance, TensorFlow, this is one example of the kind of parameters you can control to run the network. But in case of a Keras, there are less number of parameters. So for the quick training, to get a quick orientation of what this is all about, that may be a better option. Okay. So before you start AI project, as someone mentioned earlier today, there are two types of AIs. That is machine learning that depends on the supervised learning and unsupervised learning. A lot of the work we do in radiology has to do with the supervised learning, with the classification, or at times regression. So if you want to pick a package of software, software language, operating environment, and see whether they have a reasonable amount of documentation. Some of the most interesting packages came out recently, don't have much documentation. So that will be difficult to use, but sometimes it has some very interesting ideas. And try to create at least find out a community. I don't think there is a AI open source software community for radiology. And I think time has come to consider such thing because these open source packages are not properly optimized. This community is very important. In some cases, community can be 10, or it could be 20, or can be 100. But these are this community oftentimes consists of people with complementary expertise. At Ocera, we organize different committees. One community, you know what? This community came and said, we want to collaborate and we want your help. Well, okay. I said, what would you like to do? And they looked at us, no, what does Ocera want us to do? I said, no, you are the expert. A community manager like Ocera, we're the facilitator. So you decide what it is that you would like to do, decide the priority, then we will facilitate meetings, we'll facilitate the software development process. They looked at each other for a year. Finally, they realized it's their business that we are going to support. So once the collaboration took place, they realized they have complementary expertise and so on and so forth, then the productivity took off. Okay, so, well, okay, so you pick a software package you want to play with. Then some of the things you should consider is this package really optimal for diagnostic imaging? The package is stable, or how efficient is that? Because some of the packages require tremendous computing horsepower. You may not want to do that. Okay, then when you look at, let's say, convolutional neural network, how many layers can it support, and how to label images, all those things that we've been talking about today. Well, most of the open source code that's available are very good at identifying lions and dogs, but that's not really that good for grayscale images. So this is where additional innovations will be added into existing framework. Okay. And also in commercial business, it's important to know whether that's P or Q or B or D. In medical imaging, orientation doesn't really matter, I don't think, okay? So perhaps the kind of capabilities they're trying to optimize to handle characters may not be the ones that you need to pay attention to. Okay. So when you try to optimize your project, you have to pay attention to two things. We heard a lot about the data for training, data for testing, and you really have to marry the data sets you have with the capabilities that you will eventually develop on the convolutional neural network. So you have to look at the efficiencies and so on and so forth. Oftentimes people ask, how many images do you need? Okay, so everybody has their opinions. So here I'd like to just tell you one specific example of the project we did. We did the, using the lung cancer screening data, we developed a product for our partner. In that, we used about 1,000 CT cases for training and 300 CT cases for testing. And we had to add additional cases to the mix so that you can have a reasonable spectrum of cases. When then software was tested in another country, well, this country had other type of lung diseases that we are not aware of. So the software did not really do well in that country, but it's doing well in the United States. So these are some visual examples of what happens once you feed something into this black box. The number of steps it goes through. Each step has certain parameters you can control. Again, in some cases, those parameters have more to do with the computational efficiency than the computational accuracy. Okay. Earlier today, we talked about difficulties in reproducing certain research results. This was the paper published in 2017, not about convolutional neural network or machine learning. It's about fMRI. Reviewers looked at, the author looked at about 24,000 fMRI results, and that time they claimed more than 80% could not be reproduced. So that created a huge scandal. Then later it was corrected and saying that perhaps about 3,500 papers, the results of the papers could not be reproduced. So the second paper concludes their argument by saying the ease of analysis afforded by some of the software programs belies the complexity of the methods. The ease of use does not release experimentalists from their responsibility. of valid finding. I think this machine learning, artificial intelligence, I call it almost intelligence, has a tremendous potential, more than any other technologies that came into radiology, from digital imaging and all these other things that we've done. But this is going to be different. At the end of the day, if it's going to be used largely to improve the efficiency of entire operation, including perhaps outside of radiology domain, it's going to take a bit longer time. But I think the radiology community, is better prepared to deal with this because you've been at it longer than anybody else. So thank you very much for your attention.